Welcome to this brief introduction to why the heart and cardiovascular system is designed the way it is. If you are watching this video, you obviously want to learn how to fix a damaged heart and cardiovascular system. But to do this, you must understand why the heart and the cardiovascular system are designed the way they are. And further, to do this, you must realize that the heart must serve the needs of the body, and the needs of the body are served by the microcirculation. But to do this, the heart must deliver adequate volumes of blood through the arterial system to the microcirculation in a safe and efficient way. And the heart must be capable of draining all of this blood from the microcirculation through the venous system, also in a safe and efficient way. So before we can understand cardiovascular physiology, we must understand, first, the magnitude of the body's metabolic demands, and secondly, we must fully understand the very real benefits, but also the serious hazards of having such a large microcirculation. Thank you. In an earlier video, we discussed the metabolic demands made by the 37 trillion cells in an average 72 kilogram human. Now, we must consider how our cardiovascular system chooses to meet those needs. We rely on our brain for everything, and our brain relies on the cardiovascular system for absolutely everything. Humans, we are alive. But we do not make our own food like plants. We're not autotrophs. We have to eat plants and animals just to survive. We are heterotrophs. So during our evolution, we had to find our own food, and we had to evade the predators that wanted to put us on their menu. Our cardiovascular system has evolved to allow us to survive, not just to survive the activities of daily living, but to survive the extreme increases in activity, to avoid or escape the much bigger, faster, stronger heterotrophs with which we shared the African savanna. Consider the average 72 kilogram human. We are composed of 37 trillion living cells, and each of these cells depends on the function of thousands of enzymes to stay alive. These enzymes function within a very narrow window of pH, temperature, cellular osmolarity, and blood acid-base balance. And for optimal function, we require the absence of end product inhibition, that is, the absence of toxins. In addition, each cell has significant and continuous demands for oxygen and nutrients, such as glucose, free fatty acids, and amino acids, the latter producing a variety of potentially toxic metabolites: carbon dioxide, hydrogen ions, ammonia, urea, adenosine, lactic acid, and of course potassium. The average 72 kilogram individual at rest requires approximately 250 mils of oxygen per minute and produces and eliminates 200 mils of carbon dioxide per minute. Life is indeed very acidogenic. We eliminate 50 to 100 milliequivalents of acid per day as hydrogen ions, about 100 milliequivalents of potassium, and as these can only be excreted in solution, we inadvertently lose at least 1.5 litres of urine, which takes with it the precious 70 milliequivalents of sodium. Delivering and eliminating all of these ions and molecules to and from each and every one of these 37 trillion cells, while maintaining incredibly tight control of the electrolytic and acid-base physiological milieu inside the cell, is the responsibility of the cardiovascular system. To deliver all of this, then, to the kidneys. But this is only part of the story. We fragile humans, we're not particularly strong, we're not particularly fast, we do not have big claws, and we do not have sharp teeth to help us in a fight. How such an unimpressive, very medium-sized mammal survived on the plains of Africa for 200,000 years would be very hard to explain, except for our secret weapon. We could not have done it without an amazing brain, and our secret weapon would be nothing without an astoundingly effective cardiovascular system. We are alive because of the superb capabilities and adaptations of our cardiovascular system, and this is a brief introduction to the demands our body makes on our cardiovascular system. It is why we are alive. The human brain is almost certainly the reason that this otherwise undistinguished primate has continued to exist. But it is of course both a blessing and a curse, because the brain is very expensive. The human brain is responsible for a hugely disproportionate burden on the cardiovascular system. Welcome back. For the tissues of large multicellular animals to successfully replicate the early conditions of abundant oxygen for all their cells, their capillaries must deliver oxygen molecules in solution to within approximately 50 microns of all actively aerobically metabolizing cells. This diffusion distance can be greater if the cell has low metabolic demands for oxygen, but it must be much less in those cells with high metabolic demands. Ideally, for good oxygen delivery to the tissues, all cells should be within about 30 microns of a capillary, but in some tissues it's as much as 70 microns. Capillaries are 7 to 9 microns wide to accommodate the erythrocyte, which is about 8 microns diameter. Further, the cardiovascular system must deliver a total of 250 mL of oxygen per minute. Therefore, the capillary bed must have an extremely large surface area. To deliver 250 mL of oxygen to within 50 microns of all cells requires all the capillaries in the human body to have a total surface area of approximately 6,000 square meters. Given the cylindrical geometry of the capillary, this can only be accomplished if there are millions of capillaries, and this would require many hundreds of thousands of arterioles to supply them. Therefore, the tissue capillary density is approximately between 200 and 2,500 capillaries per square millimeter, and each capillary is 250 to 1,000 microns long. But some capillaries are actually as big as 5 centimeters long in specific tissues. The total length of all the capillaries in a human body is approximately 60,000 miles. Can you believe it? The intercapillary distance is approximately 50 microns, and this dictates the capillary density on average of 400 capillaries per square millimeter. An extreme example of high capillary density is the myocardium. Myocardium is, of course, continuously active. There is a capillary every 20 microns, and this requires a capillary density of 2,500 capillaries per square millimeter. Myocardial capillaries have been estimated to be 4.5 to 7 microns diameter, so the red cell has to deform quite a bit to get through. And myocardial capillaries are 500 to 1,000 microns in length. The capillaries, however, only carry oxygen close to the cells. Oxygen in solution still has to make the journey from the capillary lumen to an individual cell's cell membrane. This not only occurs by diffusion, but by bulk flow of tissue fluid, that is, convective flow of well-oxygenated plasma out of the capillary and into the interstitial fluid. And this requires each capillary bed in a specific tissue to have a very specific degree of leakiness. Let's move on to the arterial system. Welcome back. Let's talk about the problems faced by the arterial system. The arterial system must be able to meet the metabolic needs of the tissues. Little blood should go to inactive tissues, lots of blood should go to very active tissues. The arterial system must distribute blood flow appropriate to the metabolic needs of the individual tissues. The needs of the heart are also important. The arterial system must perform its function without imposing an excessive amount of work on the heart. The arterial system must meet the needs of the tissues without making undue demands on the heart. So let's enumerate the problems. Problem 1. The arterial system must conduct blood from the heart to the capillary beds throughout the body rapidly. Problem 2. The proximal arterial system must maintain a high enough pressure to drive blood through the resistance component of the arterial system. Problem number 3. The proximal arterial system must maintain this high pressure without arteries rupturing. This is a hermetic property, that is, the ability to maintain a leak-free seal. Problem 4. Blood flow to a tissue must be coupled to the metabolic needs of the tissue. Arterial blood flow must be appropriately distributed. Inadequate blood flow leads to tissue ischemia, and excessive blood flow is wasteful to the physiological economy of the body. Problem number 5. The heart generates an intermittent pulse of blood alternating from a high pressure in systole to a low pressure in diastole, but the microcirculation requires a continuous flow of blood at a consistently low pressure to avoid capillary rupture. Therefore, the arterial system must convert this intermittent pattern of high pressure blood flow into a continuous pattern of capillary blood flow. Problem 6. The central arterial system must accept blood from the left ventricle while minimizing the workload on the left ventricle. Problem 7. Some tissues, such as left ventricular myocardium, require a high diastolic blood flow because it is only perfused in diastole, not in systole. A high diastolic blood flow requires a high diastolic pressure. That is, diastolic pressures must be augmented for left ventricular myocardium particularly. Problem 8. The arterial system should guarantee tissue perfusion of vital tissues independent of fluctuations in systemic blood pressure. So, in summary, the arterial system has conductance properties, hermetic properties, distributive, resistance, autoregulatory and elastance properties that it has to have in order to meet its obligations. So let's consider how the arterial system adapts to overcome these problems and challenges. Problem 1. The arterial system must conduct blood. It must be a conduit from the heart to the capillary beds throughout the body, and this conduction must occur rapidly. The arterial system has evolved an extensively branching system which allows capillary beds to be supplied in parallel with each other, in contrast to those capillary beds which are perfused in series. The arterial system is an efficient conductance circuit, and compared to a series arrangement, this arrangement of parallel circulations and high hydrostatic pressure ensure blood reaches most of the capillary beds very rapidly. But unfortunately, not all of them, and we'll revisit this in subsequent presentations. Problem number 2. The proximal arterial system must maintain a high enough pressure to drive blood through the resistance component of the arterial system. One determinant of blood pressure is systemic vascular resistance, or SVR. The arterial system is capable of marked vasoconstriction and vasodilatation to modulate the systemic vascular resistance. In health, arterial vasoconstriction can maintain a hydrostatic pressure high enough for tissue blood flow. This is mediated by the sympathetic nervous system, and this is due to the resistance properties of the arterial circulation. Problem number 3. The proximal arterial system must maintain a high pressure without rupturing. This is one of the hermetic functions of the arteries and the capillaries. The ability of the central large arteries to resist a high hydrostatic pressure without rupturing is a particular challenge. It is due to the incredible strength of the collagen in the walls of the arteries. Interestingly, a fiber of collagen is stronger than a fiber of stainless steel of the same cross-sectional area. In health, arterial vasodilatation can reduce hydrostatic pressure to prevent arterial rupture. Problem number 4. Blood flow to a tissue must be coupled to the metabolic needs of the tissue, and this phenomena is called flow metabolism coupling. Now, this is an incredibly important concept that you must be familiar with. Flow metabolism coupling, FMC. Now let's move on to the venous system. Welcome to this new section on the venous system. Let's consider the problems facing the venous system. Each region of the cardiovascular system is a conduit connected to two other regions, one behind and one in front of it. Any region of the cardiovascular system must serve the needs of both its proximal connected system and its distal connected system. So, the venous system must serve the needs of both the capillary beds that drain into it and the right heart that empties it. The venous system must be a low pressure conduit. The pressure in the small venules must be less than the pressure in the distal end of the capillary to allow drainage. Now consider the right heart, which needs to generate a steady cardiac output. It will need a stable flow rate of venous blood back to the right atrium, and it will also require a steady filling pressure or central venous pressure, CVP. Blood flow out of the microcirculation is highly variable, so both venous return and CVP could also be highly variable. The venous system takes a variable peripheral venous pressure input and converts it into a much more steady central venous pressure output. Now, let's systematically list all the challenges facing the venous system, and you'll see them in the accompanying diagram. Problem 1. The venous system must be a low-pressure conduit, as we have said, in order to drain the capillary beds. The venous system must guarantee a low venous pressure for capillary blood to enter the venous system easily. That's the obvious one. Problem 2. The venous system must have a very low resistance. For a large volume of blood, say a total of 5 litres per minute of blood, to travel from the capillary beds to the heart rapidly means that the venous system resistance must be very low. Problem 3. Venous flow must be unidirectional, that is, antegrade or centripetal, and these are terms you'll hear during these presentations. Thus, the distal venous system must contain functional valves to prevent or at least limit retrograde blood flow. Problem 4. If blood flow into the venous system suddenly increases, or the CVP must not increase any further, the venous system must have the volume available to store this excess inflow temporarily. So the venous system needs a variable capacity to accommodate a variable increased flow from the capillary beds into the veins, but without a significant increase in peripheral venous pressure, which would of course impair capillary drainage or unnecessarily increase CVP. Problem 5. If blood flow out of the venous system suddenly increases by, say, increased cardiac output, the venous system must have a reservoir of blood volume available to supply the needs of the increased outflow for at least temporarily. So problems 4 and 5 dictate that the venous system has a variable capacity to accommodate dramatically increased or decreased blood inflow or outflow for at least a short time. A potentially large availability of storage volume in the venous system is essential to its capacitance functions, and this is true of both the systemic and the pulmonary venous systems. The former, the systemic venous system, contains 66% of the entire blood volume at any one time, and the systemic and pulmonic veins together at any moment contain 70 to 80% of the entire blood volume. So let's talk about capacitance functions. If there is a discrepancy between blood flow into and out of the venous system, the surplus or deficit must be accommodated without an excessive increase or decrease in peripheral venous pressure. An increase in peripheral venous pressure could compromise oxygenated blood reaching the capillary beds, or a high venous pressure in the peripheral veins will decrease venous return from the capillary beds and ultimately decrease cardiac output. So if the pressure in the peripheral veins is too low, this will decrease venous return and decrease cardiac output. So the veins have a large capacity, and they are referred to as capacitance vessels. Veins provide a variable volume reservoir which can increase or decrease in volume with little change in peripheral venous pressure. Brief periods of excessive venous outflow can be supplied from blood stored in the reservoir. Brief periods of excessive venous inflow can be accommodated by expanding the reservoir. Here we see a conventional schematic of the systemic circulation, and in this diagram we see one illustrates the storage reservoir capacitance functions of the venous system for the rest of the cardiovascular system. And this leads us into the next topic. How does the venous system convert a variable input into a predictable output? Filling the right ventricle requires energy to move the right ventricular walls from their end systolic position to their end diastolic position. The right and left ventricles require potential energy in the form of a hydrostatic pressure to fill it. This energy comes from the central venous pressure, that is the CVP. Under conditions of stable cardiac output, the CVP needs to remain fairly constant, but the rate of blood entering the venous system from the capillary beds is often very variable. Thus, the peripheral venous pressure is very variable. If venous filling pressures varied linearly with venous volume, as shown in this hypothetical graph, filling pressures would be inadequate at low venous volumes. But this does not happen. Why is this? Let's consider the physiological basis of capacitance functions. Consider the stretch relaxation properties of the vein wall. Elastic fibers are found in the walls of the veins along with collagen, and compared to arteries, there is only a modest amount of smooth muscle. The elastic fibers branch, interconnect, and interdigitate with their neighbors. Under tension, this network of elastic fibers can increase its original length by 50% again, and when released from pressure, they can return to their original length automatically. This property of the veins is called passive stretch relaxation and contraction. If the pressure inside the vein increases high enough, the elastin in the vein wall will stretch without any nervous system intervention or help, and the pressure within it will decrease. Conversely, if the pressure inside a vein decreases, the elastin in the vein wall will shrink and recoil, again without nervous system intervention, and the pressure within the vein will increase. However, passive stretch relaxation on its own cannot modulate venous capacity to the required extent. Something else is needed to help. And so, we'll talk about the neural pathways of the sympathetic nervous system, which also help modulate the volume of the veins. When the cardiovascular centers in the medulla oblongata are notified of hypotension by the baroreceptors, they initiate important responses. Systemic venoconstriction is one and is very important. The sympathetic nervous system centers in the hypothalamus send descending fibers in the reticulospinal tract of the spinal cord to synapse with the cell bodies of the spinal sympathetic vasomotor nucleus in what we call the intermediolateral horn of the spinal cord. These send an axon, which is the first sympathetic neuron, called the preganglionic B fiber, from the spinal cord into the ventral root into a spinal nerve. It travels beyond the sympathetic chain, enters a sympathetic ganglion as the white ramus communicantes, where it synapses with a postganglionic nerve fiber called a C fiber, which leaves the sympathetic ganglion in the grey ramus communicantes. It rejoins the spinal nerve again as a peripheral nerve to its target end organ, which for our purposes is the venous smooth muscle in the peripheral veins. This sympathetic nervous system fiber controls venous tone. Now the first graph shows that increasing sympathetic innervation or tone causes increasing venoconstriction. Most venoconstriction occurs as the sympathetic nervous system innervation increases beyond 4 impulses per second, which is essentially 4 Hertz. Arteriolar constriction is very similar, but is right-shifted, and higher degrees of sympathetic nervous system tone are required to produce arteriolar constriction, although the threshold varies with different regions of the arterial system. As sympathetic nervous system increases, venoconstriction decreases venous volume, decreasing vascular capacity. This effect of sympathetic nervous system stimulation on veins has been demonstrated quite convincingly using limb plethysmography. Stability of the central venous pressure requires sympathetic nervous system modification of venous tone to match venous capacity. I refer to increases and decreases in venous blood volume to venous vascular capacity to control CVP as venous volume to vascular capacity matching. If the venous blood volume and venous capacity change in opposite directions, it destabilizes the central venous pressure, and I refer to this as venous volume to capacity mismatch. Thank you for watching.